really, really complex systems like human beings all the way to much more simple invertebrate models. And so one of the things that, kind of my own thought experiment, when I think about aging in the human sense, and this kind of actually ties back into computer science. So is to think about the trajectories of aging. So not just how long an individual lives, but how uh, good is their health at any given point of their life. And so what you're going to see here is actually from an old paper looking at modeling human aging just off of predictive algorithms used to detect what kind of essentially like an early version of FaceApp, where if we take one individual, there's a certain likelihood that they will proceed down one path of aging as they get older. But you could imagine, biologically speaking, there is a whole amount of inputs, both genetic and environmental, that integrate to cause this individual to go that path. And maybe if they had had a slightly different medication late in life, we would see a divergence in end of life outcome. And maybe a slightly different lifestyle in midlife, or potentially an accident that caused a disability, could even more drastically swing the total life history trajectory. And then we could actually extrapolate this all out and look at how interconnected this network of outcomes are, all by trying to measure the health through time at all these different points. So one of the things when we talk about measuring these health trajectories and aging trajectories is, and also, please, anyone has any questions, feel free to interrupt. I'll keep rambling and keep going, but don't hesitate to just jump in with any sort of questions. I'm really happy to take anything that comes up along the way. So, I've been interested in aging a really long time. I was lucky enough to have a grandparent that lived into the hundred and teens. And so as such, I've kind of been conducting this experiment with an N of 1 over the last about decade. So in this case, we have our subject early on in the experiment, their first year of undergrad. By the time the second year of undergrad rolls around, we've actually seen a drastic increase in the rate of aging, some sunken eyes. We fast forward 7 years, we start to incorporate more and more signs of aging, receding hairline, and then we see the toll that the environment of even just 1 year of grad school has on our subject. And well, I kind of use this as a joke about how I am an old man and getting steadily older all the time. I think this is actually kind of a really interesting concept, because the human eye can look at both of these photos of myself, and you can immediately tell which one is older versus which one is younger. And if we wanted to, we could actually go through, pull these apart, and we could look at what are the changes. So like I talked about, receding hairline, change in fitness, wrinkles, all the stuff that kind of the human eye aggregates. But we could also look at, with enough funding, we could actually look at the molecular markers of aging. So I could go get a blood draw. I could get my DNA sequenced, and we could look at, how is gene expression changing over time in this individual. But in a lot of ways, these are hard to do in humans because it takes so long. Like the fact that this one just little joke of an experiment took 7 years is longer than a lot of funding cycles. So as aging biologists, we turn predominantly to model systems. And so here we kind of have a scale of what it looks like, where we find humans living about maximally about 120 years or so. And then if we work backwards, we've got monkeys, some really long-lived rodents, more simple like uh, uh, like zebrafish models. Mouse models are really complex, but even then, they take 3 years. And so when I think about like the impact that I want to have on the world as a researcher, um, the end goal is trying to improve people's health as they age. And so, testing a single drug in a mouse model would take nearly all of a PhD, a full postdoc. It takes a long time. So we really want to run those with the best possible outcomes. And so we shift leftward on our time scale. And so you find a lot of labs like mine use yeasts, worms, flies. Highlighted in red here is a really cool novel idea that people have had for the last 5 or 6 years, looking at killifish out of Africa, because they're pretty much the shortest lived vertebrate that can be cultured in a lab, where they only live about 6 months. And so drawing us back to the hallmarks of aging, we find that you know, all of these really disparate organisms that range from invertebrates to vertebrates, you know, in this case, we're looking at, you know, just one lifespan intervention of dietary restriction. We can get the same lifespan extension pretty much across the board. And these are currently being tested in monkeys and humans. And work out of Ross Anderson's lab has really been promising, looking at how dietary restriction is slowing aging in monkeys currently. And I know this is a really heavy slide, so what I want to not look at any of the labels of the genes, but if we look across these three invertebrate models and then bouncing up to mammals, what we find in this one idea of intervention, so dietary restriction, we find really, really highly conserved hallmarks. So if you look at across the board, we find TOR as a mediating molecule for aging. And then downstream of it, we have a series of kinases. And so this is really just to cement the point that I want to, you know, bang home here, is that, you know, as biologists, we're turning towards these invertebrate models because they allow us to do cheap, quick experiments. But the insights that we take molecularly can be extrapolated out to higher level mammalian system like mice, and hopefully someday to people. Does anyone have any general questions on like aging before we kind of start talking more specifically about, you know, the work that we're doing in the Caberland lab? And we can also cover stuff at the end as well.
Sure. I just wanted to go over how how worm aging is generalizable to like mammalian aging.
Yeah. So, I mean, if you think about the evolution of organisms, a lot of the things that, you know, govern, you know, like mitochondrial metabolism, you know, predates basically the divergence between invertebrates and vertebrates. And so a lot of these things that are basically so tightly nested with just being alive are so fundamental into like how an individual cell functions, that so the metabolism that a yeast cell undergoes in which it processes energy is remarkably similar to how a human cell processes energy. You know, it's just a matter of a much larger network. And you know, there are big differences along the way, and we do find that there are some molecular mechanisms that are not conserved. You know, yeast have, you know, our lab has worked on yeast, and I've dabbled in it myself, and there's you know, specific things that govern yeast aging in terms of like how ribosomal DNA is you know, replicated, where that just doesn't happen in humans. And so we have to, as biologists, have to be very, very careful to not make the assumption that everything is conserved. But we can go through and do the genetic analyses to look at, you know, find genes that have as similar homology to human ones as possible. And functionally, you know, I think you'll see later on some of the stuff I'm going to show you guys, we do have pretty good evidence that some of our results are directly transcribable from human data. That's a great question. I hope I answered it at least vaguely.
No, that was a great answer. Thanks, Ben.
And that'd be a great question. I know Matt's talking in 2 weeks, so definitely ask him that, and you'll probably get a much more coherent, well-informed answer because he's been doing this a lot longer than I have.
Sure, I'll definitely ask that.
So the animal that I'm going to be using for this talk and kind of what we're talking about is C. elegans. So it's an invertebrate worm that is barely visible by eye, and it's been used in research, you know, for about a little over 50 years now, almost 60 years. And what's great about it is that it lives only about 2 to 3 weeks. And so we can iterate through huge amounts of generational time, huge amounts of data, all very, very quickly. And then kind of bringing it back to the point of human relevance, what excellent researchers have done, and what I'm using in the lab, are disease models where they've taken the gene for, in this case, let's say human Alzheimer's, or a protein that we know is heavily implicated in human Alzheimer's. So in this case, it's amyloid beta. And they transform the C. elegans worm to produce that protein, and it'll have some effect. In the case of what we're talking about, it's often paralysis or impaired movement. And so what we can do is then rapidly screen a whole variety of drugs targeting, hopefully targeting that gene to really quickly assay whether they're biologically relevant at treating, the function of that gene in humans. And I'll show you guys a little bit of that data later on. And so traditionally, this is kind of how C. elegans are housed, where you can see them on this Petri plate. And then, they are little worms. They have about 900, little over 900 cells. They respond to physical stimuli, to touch. They have a huge array of age-associated behaviors and phenotypes, and have been widely used for aging research because they live such short time. And oftentimes what'll happen is, to do these experiments, you have a plate that looks like this, and then you have a researcher going through and basically doing this, touching the animal to check every day whether it's alive or dead. And you can imagine that that is super time intensive, and really the rate limiting step for a lot of otherwise excellent research. And so in the Caberland lab, we've been developing a suite of robotic tools that allow us to rapidly screen. In this case, what you'll see here is one of our newest versions of the WormBot. And play this video. And it's essentially a very fancy XY plotter that has a digital camera that goes through and takes an image about every 10 minutes of every one of those wells. On each of those wells is a little bit of agar with a little bit of bacteria that the worms eat, and they live out their whole lifespan on that. We can add drugs, we can knock down different genes, and rapidly screen. So each one of these WormBots holds about 144 individual wells. So, at 30 worms a piece, we're looking at close to a little over 4,000 animals at any given time. And this is just one of our tools. And what you'll see here is what the data looks like. So what I've been this in is our top set of 3 individual wells are our control, and our bottom are our treatment. And so this is every frame is 10 minutes throughout their entire life. And so what you'll see here is red circles denote when a worm has died. And so just in a few seconds yet, we'll see the first couple of red circles pop up. And should be, if I remember right, on the bottom. And so they're up. Oh, it's on the top. So on our control, we see a couple of deaths start to happen. And, one of the things I want to iterate is this is actually, all of this data is being analyzed manually. So I am incredibly lucky to be working with a really amazing set of undergrads, like Justin and Paulo, who I think I mentioned before, as well as I think Raja is now on the call as well, has actually scored some of this data. All of this is very time consuming, but you can see here, all of our control animals treated with our solvent are pretty much almost all completely dead. Whereas if you look just at the bottom, just by eye, you can tell, while there is some dead animals, a huge amount of them are still alive, and they're moving much more rapidly. And so in a microcosm, this is kind of one little bit of an experiment, but over the last year and a half of my PhD, I've been going through and testing a wide array of compounds. And I think we're over 100 by this point. And this is kind of just a sample of what it looks like, where we have lifespan curves tracking relative to the control how these different drugs and different compounds are affecting all of our animals. And so we both do this in the Alzheimer's disease strains and other disease strains, but also our laboratory just wild isolates and laboratory wild type animals. And we've done this with both FDA, pre-approved FDA drugs that are already approved for treating other diseases, like metformin, which is a drug for diabetes, rapamycin, which is used in transplants, and also compounds derived from natural sources, and even in some cases like traditional medicine that has been overlooked in the medical field, and we're actually able to take indigenous remedies for healthy aging and test them in the system, and actually try and kind of have a more democratic approach with the stuff that we test. And we also actually get out even more data than that. So what you've seen so far is just how long each animal lives. But you can imagine, and I know everyone has different experiences with elderly family. In my case, I watched, my great grandmother live to 111, and she was super active all the way through her life, incredibly healthy. Her daughter, my grandmother, was, even as she hit her 70s and 80s, and my great grandmother was 30 years older than her, in her hundreds, she had less physical fitness because she, had kind of a more fun lifestyle, honestly, and had eaten and drank more her whole life. And so we can look at how healthy a person is in addition to their age, because you can imagine, uh, the let me see if I can get a battery pack down here. Hold on. Let me see. I'm going to put that in chat in case. Can anyone hear me?
Yeah.
Yeah, I can hear you.
Hello? One second. Hello? People hear me?
Yep. Yeah, we can hear you now,
Yeah.
Ben.
Okay, sorry about that. I had these charging all day. I treated myself when everything moved to Zoom, and I bought myself a nice pair of wireless headphones, and that comes with a cost of having to keep them charged. So, all right, health span, which you can tell I probably could work on, given I'm a little bit out of breath from running upstairs to grab a battery pack. So what we want to do is actually assay that for the animals as well. So, with the help of undergrads, we go through and we actually annotate how much the animals move every day. So we look at how fast they're able to move at just their natural velocity. And you can see in this data here, our control versus treatment, we have our treated animals, and their movement looks more and more uniform as they get older, when we see this precipitous decline in the black untreated animals. And every dot here is a total amount of movement for every worm at each day of its life. So, the vast majority of the population early only move a little bit, but then we have this big spontaneous movement of very healthy, very active individuals. And we see under our treatment, we have a huge amount more of old, spontaneously moving animals into late life. And what's interesting is that we can test another drug, in this case, resveratrol. And what we see is actually a weirdly inverse phenotype, where our control animals have a decline in both overall movement and spontaneous movement, but our treated animals start with very little movement and then drastically increase and have almost a young phenotype even late in life. And so this is just a part of what we can do. Digging in deeper into data, we can look at the percent of time spent on and off food as a metric for, how well they're able to sense their surroundings as a almost neurological behavior. We can look at movement patternings, average and maximal rates as a function of health. We can look at their body morphology, growth rate, and all in the concept of total lifespan. But as you can imagine from the data that I've showed, this is something that is very time consuming to annotate all of. And what we're trying to do is move away from that and move towards machine learning and computer vision models to assist us and automate this so we're actually able to extract this all out, more efficiently, but also more accurately without human error. So, kind of where we started with this, we started doing classic computer vision, so just background subtraction. And it, obviously didn't work. Our images are too complex. We tried semantic segregation, but the processing time on that took incredibly long. So we made the decision to switch to YOLO, developed here at UW by Joseph Redman. And, one of the major strengths of this is it saves a whole lot of computing time when we have, thousands of images being produced every day, if not more, in all of our experiments across the whole laboratory. And so YOLO divides every input image into a series of grids. It computes probable bounding boxes and bins them according to class and gives us potentially what we expect to see. And for the most part, a lot of our stuff is just, is it a worm or is it not a worm? It's like the hot dog, not hot dog thing from Silicon Valley, essentially. So what you'll see initially is some of our first work on it. So this data set was developed all through training from undergrads using a MATLAB interface. And we only actually gathered about 470 images total. We've now increased that and as we've all been kind of stuck at home and had undergrads that are willing to do this and help out remotely since they're not allowed in lab right now, we've increased that to about 2,000 images now. And our initial data set had about 13,000 worm bounding boxes. And so, what you'll see here is, it's identifying the worms. And then even our early attempts are actually proving very, very accurate with, minimal false positives in this image. You can see just 2 on the edge there that we're hoping to get around by just, automatically masking out anything that isn't the actual auger, and only a couple of missed calls overall. So this is actually, looking really, really promising, even in its early stages. And our goal is to start broadening this out and not just look at, is this a worm or not, but start tracking, starting essentially working backwards, starting in late life and seeing how accurately we can track every individual through their whole lifespan. And so on the engineering side, we're building a set of robots that have single cameras that are just focused on one well for the whole time. They're lower throughput, but they give us, data every 10 seconds, essentially a time-lapse video for the whole animal's life. And so once we have that, what's really exciting is we can start picking apart the black box of kind of the internal calculus that's governing how these animals age, and start looking at, worm A had, this type of feeding rate, it lived this long on this drug. Worm B had this type of feeding rate, but this motility pattern on this drug, and start to actually really, really holistically look at how the scope of aging is occurring. And I think it's really promising because it's going to allow us an extra step in translational kind of holistic approach for turning this eventually into mammalian data. I think that's personally really exciting. I'm going to do a little bit of a gear shift in a second here and talk about, one other aspect of how the Camberland Lab is collaborating using machine learning, but it's a little bit skewed. So are there any questions before we move on to kind of the other stuff about kind of our worm detection and tracking models?
Do you mind going back to the first couple of slides again, Ben? Unfortunately, we missed those in the recording.
In like the very beginning?
Yeah.
Yeah, of course. Let me pull up here. Like this?
Yeah.
Do you want me to show the slide or do you want me to go through it again?
You could Yeah, you don't have to say all the stuff you said the first time, just kind of a quick recap of like the first 3 or 4 slides.
Yeah, so aging is kind of the central axis of all human mortality, and most of our causes of morbidity are highly associated with aging. And so the goal is by slowing the aging process, we can end up, potentially living longer, but living much healthier later in our lives. And across most model systems, we have shared hallmarks of aging that govern molecularly the underpinnings of the aging process. And we can kind of translate this to the human idea of having different life outcomes dependent on, do you go and but from the interplay of genetic and environmental factors, which we experience as we go through the aging process, to produce from a single, kind of watershed almost, or landscape where you have pluripotent potential or semi-pluripotent potential when you're young, and as you move through in your aging trajectory, your potential outcomes become more and more narrow. And then jokes about me getting older. I think that's probably the first couple. Is there any other that you know for sure got missed?
I think it's pretty much this. Ku had a question, I think, on regarding the first slide, something about the accuracy of the model you used.
Yeah, Ku?
Yeah, I'm curious about the accuracy of the YOLO model.
Yeah, so it's it's still in its infancy, but we're getting about, our estimates are about 95 to 97% right now for our trained images. So it's highly accurate at just detecting animals, and our next steps are adding an additional layer on top of that, where we can look at, does this bounding box change in shape over time? Does it go in and out between images in the time lapse? Which, if it's going in and out, it's likely a false positive that through a little bit of lighting interaction catches something that looks like a worm. And you can look at, let me pull up, I think this is a pretty good example. You can see that when the animal moves, it actually leaves a sinusoidal pattern behind it. And so under just slightly off lighting conditions, computer vision often will mistake those for worms themselves. And normally our contrast is high enough on these images that that's not the case. It just, if someone were to bump the robot, say, on one of the images, we can end up with those false positives. And so that's where a lot of our inaccuracy is coming from.
Yeah, thanks.
Yeah. And then, the last thing I'll touch on, and I really want to stress that this is not my area expertise, and this would be another great person to invite to give a talk if you haven't already, is Professor Sue-In Lee out of the computer science department. And so, what she and some of her grad students are doing is applying, deep learning approaches to the vast amounts of gene expression data for, in this case, Alzheimer's disease. So, mapping patient outcomes and actual biological like tangles and fibrils in deceased brains with expression data from those same samples to find genes of interest that might govern the progression of Alzheimer's disease. And so what we're doing is we're using our Alzheimer's disease strain, and we're then taking the genes that they're giving us from their their deep learning approach and testing whether or not those are actually governing the progression of Alzheimer's disease in our animal. And it's in this sense, I think we're getting at really highly translational research where we can knock down the specific genes that they find and then look at how slow or fast our animals paralyze as a result of the Alzheimer's proteins. And this is some early data that we found based off one of the genes that they suggest to us. And this is the rate at which they paralyze. So the earlier in life that they paralyze, in this case the control, the quicker the disease is progressing. And then if anything that's slower than that, that's taking longer to paralyze, is something that's slowing the progression of Alzheimer's in this model. And we find that UCR1, which is a part of the metabolic machinery of mitochondria, is actually knocking that down in our worms, based off of human patient data, is actually slowing the progression of the disease in our animals. And so I want to stress that I'm on the biology side of this, not the computer vision model. And they've published a couple of great papers on this. So I'd really recommend reaching out to maybe see if they'd give a talk, and also reading those papers. But this is kind of the intersection between the biology and computer science that I think the Kaeberlein lab is really on the forefront of, which is really exciting to be. And I think she's in the call right now. Raja Estes is a former undergrad that's now continuing as a technician, and she's following up on this work. So I think I've talked at you guys for quite a while, and there's more stuff we can go into if people are excited. But I want to take a breather and let people ask questions, and also very much acknowledge all of the work, particularly by the undergrads that have contributed so much to this research, and just say, if you have any, we have total positions both at the benchtop and also doing the computer science behind all of this. So that if you feel like joining on, please don't hesitate to send me an email, and we can loop you in and get you going on helping out. So with that, I'll take questions. If you want to talk about yeast, I can show yeast data, but I think we covered most of the most exciting things that we're doing in the Kaeberlein lab right now.
I had a question about the, so earlier in the slides, you mentioned that you would basically make a time lapse of each worm's life.
Yeah.
Is the computer vision model able to like uniquely identify different worms, or how does that work?
So right now we're not. We're working. That's like actually what we're working on today, like today before this meeting. So what we're using is we're using techniques that are taken from computer vision solutions for people on tracking individuals on security cameras or tracking cars in a crowd. And so we're using the same sort of like RNN approaches to not necessarily track an individual based off its characteristics so much as infer within the time lapse of images what the most likely between two images, which worms are most likely the separate worms. And from that point, then we identify them, we draw them back. And so what we're working on then is it'll actually be really exciting, because no other lab is really able to do this in as natural of environment as what we're able to do. The only other successes that have been had with this are generally either using like hydrogel droplets to encapsulate worms in a little bubble, or in microfluidic devices, which are excellent tools but are not the natural environment that most aging research is done in. So our goal is, by being able to track them individually on these plates, we're able to take experiments that have already run by our lab and other labs and contextualize the data longitudinally for each animal within the environment that's already been tested, rather than you could imagine a drug that affects feeding behavior is going to affect worms differently if they're housed in a liquid slurry of food versus on a plate. And so hopefully this allows us to better able to connect existing data. That's a great question.
Thanks, man. That was pretty interesting. Okay, I do have another question.
Yeah.
How do you search through the space of drugs and treatments usually?
So we have a couple of ways that we do it. So, one is that there are some broad compound libraries out there where, you actually purchase them from pharmaceutical companies that'll give you, it's like, 10,000 compounds, and you can just run through them. There's people out there that also do it looking at the similarity in molecular structure. So if you know the right you know that a drug that has X combination of, you know, aldehyde and benzene ring groups in a certain conformation affects your phenotype, which for us would be aging. Then you start to say, okay, what if it had one extra aldehyde group, or what if that aldehyde group was ethyl group, or, you know, it's been a long time since my organic chemistry, if I'm honest. So, but they'll start to look at them, and there actually are some labs that are using, you know, machine learning approaches to try and dive through these big data sets of compounds to look at structurally what similarities might be underlying biological relevancy. There's also a huge amount of genetic data that's been collected from, you know, I'm right now I'm using a database of information from human cell lines. So researchers took I think 5 different human cell lines and exposed each of the lines to any of about 20,000 drugs. And some of them are just, you know, like kind of like what we test. Some of them are natural products. Some of them are pharmaceutical. Some of them are just completely synthetically derived. We don't know what they do yet. And so they measured the gene expression under each of those compounds. And so what I'm doing is I'm using that database and I'm taking our genes that we know extend lifespan, that have been positive hits either from our screens or from someone else's work, and I'm mining that database for compounds that produce similar expression effects and finding those, and then using those to start the next cycle of testing them within our robot model.
Thanks.
Yeah. And of course, people are also taking stuff from, you know, human trials, you know, stuff where, you know, someone is testing a new drug in a mouse for humans, and it's in like early phase testing, and they notice people are, you know, having an improvement in fitness on the drug versus the placebo. That feeds backwards, and then scientists like myself that work in invertebrates will then go through and do the genetic screen to see how that drug is potentially affecting the aging process. So that's another way that stuff kind of is not just us invertebrate folks feeding upward vertebrate models, but also working back and actually quantifying how these things are having an effect using our really genetically tractable tools.
Yeah, that's actually pretty interesting because I was like the reason I asked this was I was thinking about it from like a computer science perspective, right? Like when you're searching through an array, like if it's sorted, for example, you can search faster by doing like binary search. So I was just wondering if there was like something similar for like the drug search process.
Yeah, that's a good question. And yeah, I haven't thought of it like that way. That's kind of a cool idea. I know that that'd be a great question for like Sue In's group about how they use machine learning to mine these big data sets. And there's also like, you know, UW has a great genome sciences program too, which probably would have an even better answer than I could possibly come up with as well, in regards to that.
Sure. Yeah, thanks for recommending reaching out to Dr. Sue. I'll definitely make sure to do that.
Yeah, definitely. She's fantastic.
Any other questions, guys? Because I have a couple more, but I'd just like to give you all a chance to ask it. Okay, I'm gonna take that as a no, but if you guys have any other questions later on, feel free to interrupt. So one thing that I wanted to ask that you mentioned earlier, like in the first few slides, was you said you put the graph of like aging, heart disease, and cancer, and like
Yeah.
the cured, like how much they would extend people's lifespan. So I
Mhm.
was wondering, like what is like the relationship between aging and those like diseases of extreme age, let's say, like is it that if someone has heart disease when is it that someone is more likely to get these kind of diseases
Yeah.
when they're older, or is it that those diseases are more likely to affect them more severely when they're older, or kind of a mixture of the two?
It's kind of a mixture of the two. So the all of these are the risk of death from those diseases as they're related to age. And so, as we've got these molecular mechanisms of aging that are highly conserved. And sorry, my thing's being a little bit funny. And so, you can think of it where, cellular senescence is, cells, growing old, being less functioning, less able to carry signals like we see in aging brains. Mitochondrial dysfunction is really apparent in the heart because it's so energy intensive. So as these disparate parts of the biological machinery are affected by the aging process through these conserved mechanisms, at that point we see the risk of these diseases go up, as well as the severity that those diseases take, increase as we get older as well.
For sure. And so on the previous, or like on one of the slides, you mentioned that the hallmarks of aging are like conserved between species, but like, for example, like for like a bacteria, for example, it doesn't really, well, I guess that's different because it's like a single-celled organism. Is it that, like do single-celled organisms like those age, or is that really a meaningful term for them?
So yeah, single cell organisms do age. So actually, I know, I'll be honest, I know very little about bacterial aging, but I do actually know a fair bit about yeast aging since our lab works a lot with that. And so, you know, there's some aspects of it where, a single-celled organism, altered intercellular communication maybe isn't as relevant. But if you think of that single cell in relation to the other yeast in the colony, their ability to send signals between the other cells is, correlative with this hallmark. But we know that like some of the features that govern genome instability in yeast are conserved all the way to humans. And we can look at, you know, uh, genes that when mutated in humans cause cancer, they'll have a similar effect of altering the cell division rate in yeast cells if we mutate them. Similarly, mitochondrial dysfunction, we can look at the conserved mutants. And so, one of the things I didn't necessarily get to because I didn't want to like I felt like I was throwing a lot at everyone, but we use yeast as a model system within the lab where we okay. We basically trap single yeast cells over time in these microfluidic devices, and then we can label single genes within the cell. So in this case, we've got in red, that's the nucleus of the yeast cell. And in green, that's some of the machinery that when a cell divides governs how it divides. And so we can track those in real time. And so I think one of my other students, Justin, is on this call, and he's working right now to basically take all this data that we currently have and start mining it, where we can look at how many times a yeast cell divides as its age, and then start to pick apart through all of this essentially demographic data of yeast cells, how is one gene or one intervention governing their division rate. And in this sense, we're almost using yeast cells as a model for human stem cells, because they're asymmetrically dividing cells. So stem cell, just keeps on producing, semi-pluripotent cells until it senesces and dies. And yeast are mother cells are the same way, where they keep producing new cells, but as the mother cell gets older and older, it starts to produce more and more damaged and almost pre-aged, old-looking daughter cells until they both die. And so, like we talked about, you have to be more careful as a biologist when you make assumptions with these as you get further and further away from humans. But we have established proof that a lot of these are governed are similarly patterned. And actually, this is a great example of it. That this would be great stuff to ask Matt about, since a lot of his early work is foundational in this. Where this molecule called TOR that governs how dietary restriction is mediated in both mammals and yeast was really developed, like the knowledge of that was strongly developed by Matt Kaeberlein as he was a PhD and postdoc. And we can see how the same genes similarly function, have a similar network in both model systems.
Pretty interesting. I wonder though, like, what's the difference between like a single-celled organism like yeast and like the germline of like a species? Because the germline of a species is like it keeps replicating for like billions of years, and like what is in place that keeps that like going on, but like for a yeast cell doesn't, basically.
Right. I mean, you could think of yeast cells themselves as a germline, right? Functionally, they keep producing and they've been going for billions of years as well over time. I think that's a great point. And I think dissecting out, like, there's some cool work that like is being done by people that are taking like a systems biology of aging approach that are applying whole range of mathematical tools to try and map like these relationships out. Unfortunately, I am not one of them. So I don't want to like assume too much and say the wrong thing, other than checking out the work by like one of Matt's kind of contemporaries and collaborators here at UW that also works on the dog aging project is Daniel Promislow. And so he works predominantly in flies as well as doing the demographic stuff in dogs to take a systems biology approach to these same set of questions. And it's fascinating work. It's really, really cool. If you're interested in having more aging people in your group, he'd be an excellent person. And he's a biostatistician, so he actually is probably more computational science relevance than someone like myself that's learning it as we go.
For sure. Yeah, I'll definitely reach out to him. Thanks for the recommendations, by the way.
Oh, happy to. Really, I think honestly, I'm super impressed with your guys' group. And again, if anyone is ever interested in coming on board and applying computer vision tools, or even whatever you could bring to the table, we have I've a set of computer science students there right now that are working on building a database for all of our data that we can then use to mine out and send it to bioinformaticians down the line. So like we have pretty much, if there's anything you can think of doing as a computer scientist, we have room for people to get involved in lab and get a letter of recommendation from Matt.
Nice.
Yeah.
Are there any qualifications you would like for those kind of positions? Because I could definitely make a post about that on my club page.
Yeah, so we'll happily take anyone, right now, experience with, YOLO or any, computer vision, it would be incredibly helpful. A big push of what we're trying to do is migrate our data from like local servers onto Amazon Web Server and move our analysis tools as well to get hosted on AWS. And I am very ignorant of how AWS, honestly, the fine details of how it works. So, if you know any bits of SQL or anything like that, we definitely have room for people to help out as well. And yeah, those would be the two main features, is basically database work and then computer vision. We also, if people are on the engineering side, we have a whole host of, engineering problems with the WormBot itself that we're trying to solve as far as programming robotics, how to, control them effectively, collect data and organize it automatically. So, but yeah, in computer vision and YOLO neural networks, then database science would be fantastic. And feel free to post my email if you make that post, and I'm happy to review it or review. I'll happily take any applicants.
For sure. Yeah, I'll make sure to do that.
Oh, I really appreciate that.
Yeah, it seems pretty cool. I'm glad to help out if I can.
Yeah, and you know, I know Matt's talking to you in a couple of weeks, so I think you'll get, you know, you've had the trainee talk, and you'll see the master.
Right.
So
For sure. And just as a preface to his talk, like what is the, I know there's like many different things, like variations genetically between species, but like, is there like a primary like difference between different species on like their lifespans, or is it more like a complex variety of factors?
So that's a great question. And that's something that people are actually trying to say. It's, you know, there's some stuff that is conserved. So let me find out where I've got my slide. And so, you know, this is actually a perfect example of that. So if we think of like, you know, a genus, I think, yeah, genus, rodents. Is that genus or is that something else? Does anyone know?
I'm not sure, honestly.
Class, family, yeah. So, you know, broad category species, rodents. And so if we look even on this infographic, we have, you know, this mouse, which is a rodent living 3.5 years. And then, you know, bats and naked mole rats can live, you know, almost 40 years. And so even within these very similar species, there's a huge range. And then if you look at, like, you know, one of the things that Matt will probably talk to you guys about is how, you know, the data that they're getting from the Dog Aging Project, where you have, you know, canines, which are it's an even smaller category. And, you know, you've got, you know, what Scooby-Doo is a, not a greyhound, not a mastiff. What breed is Scooby-Doo, guys? Come on, someone knows
Great
this.
Dane.
Great Dane. Thanks, Raja. 5 points. So Great Danes, you know, live like as short as like 5 years, maybe 6 years on average. I think I could be a little bit low, but yep, they live incredibly short. And then on comparison, you know, you have species that, or, you know, breeds of dog that live, you know, and it's generally smaller ones too, that live up to like, you know, 14 or 15 years on average. And so examining those differences and trying to figure out what specifically is genetically responsible for it is one of the principles of the Dog Aging Project. And so, you know, we don't necessarily have an answer. I think, you know, Matt will probably say that, you know, size is a pretty good indicator. Generally, larger breeds live shorter. A recent PhD graduate from my program that actually worked for Daniel Promislo, Kelly Jin, she published several papers on, you know, mining demographic data from dogs to look at what physical characteristics might underlie some of these changes, even within breeds. And, you know, size was the one that really stood out as being larger dogs generally live a little bit shorter. But it's a fantastic question, honestly. And I think we still don't 100% know.
Yeah, the size thing is interesting, but then you have obviously there are exceptions like the whale and stuff like that. So yeah, I'll definitely make sure to ask Matt about like what the theories behind like the relationship between size and lifespan are. It seems pretty interesting.
Yeah, no, that's it. I'm curious. I might actually tune into his talk and see if he brings it up before you ask it, because I know he's mentioned it at some other talks and gets at it a little bit early on. So I'll be really curious to see if he gets ahead of you there.
For
It's
sure.
a great question.
Did you have any other slides you wanted to share with us? Maybe even just briefly.
We actually covered just about everything, if I'm honest. So we kind of went through it all. So, you know, really, you know, feel free to shoot me an email if you have any other questions. I'm happy to answer anything. We could even do a smaller Zoom meeting if anyone is interested in joining the lab or interesting. And yeah, we've got a great team. Huge amount of undergrads have won Mary Gates Awards in the lab, 3 of which have worked just specifically for me in the last year and a half or so. And yeah, thank Pete, who's around here somewhere, but not with me here in the basement right now.
Nice. Thanks for coming, Ben. Really appreciate this. It was very interesting.
Hey, thank you so much for the invitation and for organizing this. It was a real treat to get to talk to you guys. And yeah, look forward to maybe getting contacted by some people that are interested in aging.
For sure. All right. Thanks for coming, everyone.
Take care, all.